Hi, welcome to the Bioinformatics Chat. Today I'm talking to Aleksandra Kolodziejczyk. Hi, Aleksandra.
Hi, it's nice to meet you.
Nice to meet you too. So you are a postdoc at the Weizmann Institute of Science, and it seems that you did quite a bit of work in single-cell RNA sequencing. Tell us a little bit about yourself, about your background, and how you got into biology and bioinformatics.
So I graduated with biology degree from Italy, actually, in Perugia, and then I did my master's in molecular biology in Heidelberg in Germany. And then it was obvious for me to pursue a PhD, and I joined the lab of Sarah Teichmann in Cambridge. And I was actually supposed to do something a bit different. So it was still in 2012. And when I applied, it was 2011. So it was before the single-cell RNA-seq days. I was supposed to do chip sequencing. But when I arrived, the first project with single-cell RNA sequencing kind of appeared. And I started working on that because I found it very interesting. It was a very new field, a very fast pace. We were one of the first in the world to do that. I was the first one to have a single-cell data set from the Fluid MC1 in UK, which was really great. So I kind of committed to this topic through my PhD for a full 4 years. And now in my postdoc, I'm also working on genomics. Well, single-cell sequencing is a big part of that as well. So I was doing both experimental part and the analysis.
Very interesting. And you mentioned something that I also have this impression that single-cell sequencing is something very recent. Although when I read papers, I see that actually single-cell sequencing, or at least some kind of single-cell analysis, have existed for a very long time. But it seems that it really took off in the recent years. Why do you think this is so?
Well, so the first paper about single-cell sequencing with just like couple cells was from 2008 from a lab in Cambridge. And then there was another one in 2009 by the same group. But only in 2011 and 12, the chemistry that allowed to sequence more cells with reasonable price kind of appeared. There were a couple competing methods. And basically only from this time, the whole field flourished and boomed. So it was around like 2013, 14, 15, when everything started to develop. I mean, we couldn't really do single-cell sequencing much before because there was no sequencing before, like RNA sequencing.
Yeah, that's true. Perhaps I was thinking more about like general single-cell analysis.
Well, there were other things. There were some trials of doing single-cell microarrays, but this didn't really work out because of the sensitivity issues, probably. There was single-cell qPCR, which is method where you have to decide on which gene you analyze. And this was known already since a while. But I mean, single-cell approaches which involved microscopy or flow cytometry were known for like years. And mostly people who are working in immunology and they use a lot of flow cytometry. That's how they approach things for a really long time. But the important difference is that with those old methods, you could only measure one gene or one protein at the time, or maybe 5, or maybe 8, but not much more. And with sequencing, we measure all the different genes in one cell, and that's really useful.
Okay, so now that we have this amazing technology, what can we actually do with it? So I know you yourself did a few studies using this technology. So what interesting biological problems can you address with this?
Well, there are several kind of groups of problems that we can address. I mean, in the beginning, it was just a tool to look at situations where you have a single cell. So in the beginning, in the early embryo development, you just have one cell, and then 2, and then 4. So it is not possible to do sequencing in en masse with the bulk approaches because you don't have a bulk. But that was the beginning. And then people realized that this could be a way to survey a population of cells. So within some populations, for example, in immune cells, there are many different subtypes, or in brain as well. And sometimes we know all of them, but we could never be sure because the methods that we used were methods involving only measuring a couple genes at a time. So this method is used to kind of understand what comprises the population. So you can take this sample from the blood, from the brain, or wherever, and see what is there, and you can identify new cell types or new cell states. And these things are kind of obvious and easy to do, but then the whole technology was kind of taking one step forward, and we understood that we can also look at different functions. So if the system is somewhat perturbed, so for example, you add some stimulus and some cells will respond, others will respond a bit less, you can do correlation between genes and you can look at the regulatory networks. You can look at the process of, you can order the cells along the process. We call it along the pseudo time. And this is also very useful to study some dynamic changes from different cell states. We can see if there is some intermediate, which is stable and things like that. It's very useful for studying development. For example, development of blood cells from hematopoietic progenitors to different blood cell types that we have. It was also used for some other things, like myself, I used it to look at the monoallelic gene expression. You can use it to look at the cell cycle, what we also have done. So there are many questions. So it's, for example, with the cell cycle is very cool because what you can do is you put your cells in the sequence them, and then without any other knowledge than the transcriptome, you can know which cell comes from which cell cycle phase. And so you can sort them, and you can even estimate the speed of cell cycle from this data. So this is very useful. And surveying the data can give you a lot of information about your population of interest.
So let's talk a little bit about cell cycles. First of all, like on what scale are we talking about?
I mean, the time scales here are very different. I mean, the short cell cycles in stem cells that I was working with were around 12 hours, let's say. But mostly it is way longer. And if cells cycle very quickly, then what you observe is in RNA is that different stages of cell cycle are not as discrete as if cells cycle longer. So what you observe is you have a cell and it will express, as, for example, particular genes that are specific for one cell cycle stage, but it will also express a little bit of the previous because the degradation of the previous one didn't happen because the degradation of these genes is quite slow. So they have kind of leaked from the previous cell cycle stage. And this allows to understand the speed. So if there are very discrete phases, that means the cell cycle slowly. But if you have this kind of spills from the previous ones, it means that it was fast because they didn't manage to degrade. And this is really useful because, for example, if you imagine you have a cancer and you have different population of cancer, different clonal populations, right? Because cancers are very heterogeneous, and you see that you have, let's say, three of them, and you see that one of them is cycling way quicker than the others. And then just from this one single snapshot of the data, you can tell that this one is growing faster, even though you don't look through time. And I think this is pretty powerful and pretty cool.
So let's say you convinced me, and I think this is very powerful, very cool. I want to do something like that myself. What is the mechanics of a single-cell RNA-seq experiment? How do I go from a tissue sample I have to something that I can perhaps send to a sequencer?
Well, this process can be quite easily divided into several steps. And the first step, which actually is one of the most tricky and most difficult steps, is to have a single cell separated in some way. So you need to first obtain a single cell suspension. So you need to use different enzymes or mechanical techniques to dissociate your tissue. If you work on blood, you're one step ahead because blood is naturally single cells. And then you need to obtain them as a single cells. And there are different methods here. One is you can use a sorter to sort single cells into well plates, which is quite easy, and almost in every institute sorters are available. So that's you don't need to buy additional equipment. Then the other method is this Fluidigm C1 machine that we used to use in Cambridge. And it has advantages because it is a microfluidic device that allows capturing single cells. But afterwards, you can still look at the microscope so you can see what cell looks like, what each of the captured cells looks like, which is not possible with flow sorting. And then the newest technology that is allowing us to capture way more cells than with these two previous ones is droplet microfluidics. So what you do is you have your cells in suspension and you encapsulate them in droplets in the lipid phase, and you can encapsulate thousands in a very easy way. And then once you have your single cells in whatever method, what you need to do is you need to lyse them, and that's straightforward.
So by the end of the first phase, do I always have like a well plate?
No, because if you have droplets, the cells are in the droplets. So basically the way it works, you have a tube or some container, and inside you will have like a solution of oil, and inside this oil there will be little droplets, and in each droplet there is one cell. And on C1 they are on some Fluidigm's machine, they are in the chip, they are in little special positions in the chip, but it's not a well plate. So it's a very different way to do it.
Right, and because the output of the first phase is so different, then presumably the next phases need to take this into account, right? Because you have different methods perhaps to deal with different inputs.
Well, actually not really. There are different like technical ways to deliver enzymes or like the next steps to the one of these three different options. But chemically, the thing is the same or very similar. I mean, there are still different options for the chemistry, but like in purpose, they are very similar. So first thing you need to add is the lysis buffer. The buffer that is basically like soap, and it will break the cells. And if you have a well plate, it's very easy. Actually, what you do is you sort the cells already to this lysis buffer. If you have this droplet method, there are ways when you form a droplet actually, where the half of the drop is the lysis and half of the drop it has your cells. When the drop actually is formed, the lysis happens. So you don't need to add anything. And in Fluidigm C1, the microfluidic channels, they will like add the lysis buffer. So the cells are broken and they are still in either the droplet or in the well or inside the chip.
So you break the membrane of the cell, right? But you need to make sure that all of the cell's contents is not like spread around.
Yes. Well, it's not spread around because it's either in the well or in a droplet or in the chip. So these are within some kind of discrete containers of different sort, but basically it's very similar. It's just the way how it's encapsulated is different. And then what happens next is your RNA is already exposed and it's in the buffer. And what you need to do is you need to reverse transcribe. But RNA in the cell, it contains both messenger RNA, so the one that codes for genes, and then it also contains lots of ribosomal RNA. So to avoid reverse transcribing ribosomal RNA, we use a special primer that binds to the poly-A tail of the RNA, and it will reverse transcribe only the messenger RNAs and some long non-coding RNAs, but not the ribosomal RNAs. And this way, we don't lose afterwards reads in sequencer on this quite not so useful information.
Right. So this stage is pretty much the same as with the ordinary RNA-seq, where you do pretty much the same. But I'm wondering, because you have so many individual cells, does it make it harder?
Well, there's a difference because normally now most RNA-seq is actually done, but the reverse transcription is done with random hexamer priming, which is slightly different method. So you have like short primers that have 6 different bases, like 6 random bases, and they prime randomly in the RNA. They don't prime at the end, and you obtain the poly-A fraction by pulling it with beads. So it's a different way, but it's actually very similar. The tricky part here is that you have very limited material in the beginning, so you have to be very careful about not having RNases and not losing your sample. But chemically, like biochemically, everything is very similar. Okay,
so at this stage, you amplify the the messenger RNA molecules, right?
No, well, you only reverse transcribe. So you reverse transcribe means they were RNA and now they are DNA. So because we cannot amplify RNA because PCR works on DNA and the reverse. So once you have the DNA, there are two ways to amplify. You can either amplify by PCR, where you need to have the DNA to do it, or you can amplify it by in vitro transcription. So what you do is from DNA, you make RNA, and you can make a lot of it. And then once you have a lot of RNA, then you can go back to DNA by reverse transcription. So these are two ways.
Mm-hmm. How do you make sure that, once you reverse transcribe it to DNA, but then it looks the same as the DNA that is already in the cell? How do you make sure that that DNA does not interfere with your process?
Well, in one cell, you have only one copy. Well, two copies, right? So it's just so little that you don't really need to worry about it. Because you care about the copies, right? Because what you do in the end is you have your genes and you want to check how much of the gene you have. So if you have some DNA that comes from, and you don't care about 2 copies, you care about 50 copies, 80 copies, and so on. So if you have one more, it doesn't change anything really.
Okay. So by the end of this phase, we have this cDNA molecules.
Yes, we have amplified cDNA, and the amplification is necessary to have enough material to be able to sequence. Otherwise, it's just too little. And then you need to prepare the library. Well, and again, it slightly depends on the method, because sometimes during the amplification, you can already add the special adapters that allow for sequencing, but not always. So it slightly depends. But usually what happens, you do a library, and you can use a more custom-made method, or you can use, a kit produced by Illumina or another company that produces sequencers. Just use that.
So at some point you have to transfer from whatever you have. Like you mentioned, you may have some kind of liquid with droplets or suspension with droplets, right?
Yeah.
And you have to move from there to like a flow cell that you can put into the sequencer, right? How does that happen? How do you preserve those bubbles?
Well, you don't. If you use the droplet-based method, what happens is when in the first step of reverse transcription, you add the barcode. So in each of the droplets, there is a cell, but there's also a bead with the barcodes. And this is a different barcode in each of the droplets. So when you do reverse transcription, on top of having your cDNA, there's also a barcode. And then you can pull everything together, and using the barcodes, you can know from which RNA, like which molecules were coming from which droplet. So you can pull them together. And then informatically, once it's sequenced, you can know which comes from which cell.
And this is it, right? So once we have barcoded sequences, we can just put them into the sequencer?
Yes. Well, you have to make a library, but so basically to put things on the sequencer, you need to write sequences at the end, so the sequencing can happen, so it can bind to the flow cell and so on. But that's a standard procedure that you use for RNA-seq.
Okay, and then, of course, you need to decide how much to sequence, right? Because since you have many small individual cells, you want to have enough reads for each one. But of course, when you have thousands of cells, it becomes like 1,000 times more expensive. So you have to find some compromise there, right?
Yes. So in general, because the samples, the complexity of the library is pretty low in comparison to normal standard RNA sequencing, because what happens during this process of preparation is you only kind of only measure around 10% of the molecules. So everything that is lowly expressed, even if you have some signal, it is a little bit unspecific. Or like it's very hard to know whether 0 is really meaning that this thing is not there, or whether 1, it really means it's 1, or maybe 9 because of this problem. So what you can actually only measure is the things that are highly expressed because of this procedure, how you make it. So you don't really have to sequence very deeply. Sequencing depth will depend on your cell type. So if you have smaller cells, you actually don't have to you cannot really sequence more because it doesn't give you more information. If you have slightly bigger cells, it's better. You can sequence a bit more. So that's one thing. So it depends basically on the number of molecules that you have in the beginning. And it depends on the technique that you use. Because when you make the libraries, you can either have the fragments from the RNA spanning the whole length of the RNA, or you can do so-called molecule counting, where it only comes from the very end. And obviously, if you have the full length, it's slightly different than if you only have the very end, because there you can sequence actually slightly less. So these things you have to kind of take under consideration when you decide. But in general, something between 100,000 and 1 million of reads is a lot. Like people do something between that.
Yeah, this sequencing at the end, it reminds me, I think it was like in the early days of RNA-seq. Originally, they did that, and then they moved to like full-length RNA sequencing, which allowed them to reconstruct isoforms, right, and increase the precision. And from what you're saying, it sounds like the single-cell sequencing is maybe at the same stage as RNA sequencing was a few years ago.
Yes, I think you're right that it kind of goes through a very similar kind of development pattern. There's still like technological advances that can be made to make things better, and they are being done. So I think we will get better, but obviously it will never get as good as bulk sequencing in the meaning of precision and of like how it really recapitulates what's in the cell because of this really low amounts of material. So that's really at the edge of what we can do. Yeah.
So from the statistical point of view, I was thinking that, on the one hand, you're interested in the differences between different cells, right? Otherwise, you wouldn't be doing single cell sequencing. Uh-huh. On the other hand, there are statistical methods that allow you to what they call borrow strength between your samples, meaning that you rely on the fact that these are typically cells from the same tissue, perhaps, certainly like from the same organism. So there are a lot of similarities there, and sometimes it is possible to leverage these similarities, even if from a single cell you don't have enough signal. But averaging across many of these cells, you can synthesize some kind of signal, right, and make meaningful conclusions.
Yeah, that's true. So what people usually do is when they get their data, they will first perform some kind of clustering to see what kind of cell types they have. And then they will compare between clusters rather than compare between cells. And this obviously, as you said, it helps to gain more information because you can see that, let's say 90% of cells in one cluster expresses some gene of interest. Maybe 10%, in 10% it's not detected, but we can kind of assume that all of them express this gene and that this gene is a characteristic of this particular group. So that's a very common strategy.
Right. And I think I came across some sentiments that single cell sequencing can actually be like a replacement to the classical RNA sequencing and increase its precision, because there's this trope that whatever you get out of RNA sequencing, it is not representative of what is happening inside individual cells, because cells are so heterogeneous, right? And so if you sequence each cell, then you get a clearer picture of what is actually happening there. But on the other hand, there is this counter argument that you bring that each particular cell has very little material. So do you think, for example, today or maybe at some point, is single cell RNA sequencing like an improvement upon classical RNA sequencing, or do they have different niches?
I think they are complementary. So I think if you have a population of cells where you are not sure what cell types are there and what you can see, or you are interested in some heterogeneities, single cell sequencing is great. But for example, if you do that, you found your new cell type and it's really like great, you identified what genes it expresses, you can identify what are the cell surface markers in that particular new cell type that you discovered, right? And you can, using these markers, you can easily isolate these cells. And then you can do bulk sequencing to characterize it better, because single-cell RNA sequencing works well for highly expressed mRNAs, doesn't work well for low-expressed RNAs. There are some methods for microRNAs, but it's also way not as good as the bulk methods. And then there are long non-coding RNAs and other things, other RNAs that are lowly expressed. And for these, at least at the moment, single-cell RNA sequencing is not the method of choice. So I think it is useful to kind of combine.
Interesting. I see a lot of papers somehow published about normalization of single-cell RNA-seq data. Could you explain what normalization is and why it is important?
Well, there are many different things you can normalize there. It depends what you want to do and what kind of data you have. Because like when you do quantify the gene expression, you may get two kind of different types of data. One type of data is using unique molecular identifiers. And these are little barcodes that you add during the library preparation and so on. And they allow you counting the genes, because during the preparation of the library, especially if you do PCR, there are some PCR biases and so on. So when you do that, you actually can count the molecules, because if some of these barcodes appears 3 times, you know that it's just because of amplification, but it was just one gene. When you get these data, you have actually counts of molecules, and these are way closer to the reality than if you have some kind of more abstract, just reads from the molecules. And it's like with RNA-seq, there are different ways to normalize, right? The data people use to compare between cells, because it depends what you want to look at again. Because, for example, one cell can be bigger, the other one can be smaller. And the question is whether you want to compare the absolute number of the molecules in the small cell and the big cell, for which unique molecular identifiers would be the best option. Or you want to kind of make them to be average size and normalize and compare what would be the number if the cells were of the same size. But by size, I mean not the physical size, but rather like the total number of the molecules within a cell. So these are things that you need to look at. But then the other things that people normalize are things related to batch effects. And that's yet another big story, because most of these methods actually suffer from some batch effects, especially the droplet-based method. So these things have to be thought of.
Yeah, so you mentioned using molecular identifiers to quantify the absolute count of molecules, right? Rather than the relative count. And another technique, which I think they're both necessary, is spike-ins, right? I know this is important in classical RNA-seq, because you typically sequence only a small sample, a small subset of all RNA molecules in the sample, in the tissue. But I don't know, is this an issue in single cells? When you sequence RNA from a single cell, are you typically able to sequence everything there is there? Or is it still like a small subset of all the molecules?
Well, I think it works a little bit different. So the role of spiking is to make sure that the library has a good quality, and that what you see in the end is in each of the wells, it's at least technically the same. So that's why you add a little bit of spiking to see that in the end, all the spikings from all the samples should be the same, but the biological result can be different. And then you know that biological differences are actually due to real differences, but not to the fact that there was some little evaporation in one of the wells or something like that. So what you can do is, that's why you add the spiking to all the samples. And then once you have your data, you can cluster the samples based on spikings and see if maybe there's some batch effect, things like that. So that's why the spikings are very useful. So the complexity of these libraries is pretty low. And obviously you don't sequence all of the molecules because most of them are lost. But I think the key problem here is to ensure the uniformity of the preparation of the library.
Right. Okay. And once you get the results from the sequencer, and maybe we should talk a little bit how those results look like. So originally you just have a huge FASTQ file, which is just all the reads, right? So then you have to sort them by the barcode. And then do you, like in classical RNA-seq, you align these reads to the genome or the transcriptome, or maybe assemble a transcriptome? Is it the same?
Well, not really. Well, we don't really assemble transcriptomes because the quality of this data is not as great as of the normal RNA-seq. So it's better to align to known, to mouse or human genomes. I mean, most of the single cell data that we have at the moment are from these two models. I mean, maybe some zebrafish and maybe there's some plants as well, but mostly aligned to the genome. And the pipeline is for this basically the same as with the normal RNA-seq. What you do, you map, you count your either you count your UMIs or you count your reads, and you end up with the count table where for each cell you have number of molecules or level of expression for each of the genes.
Okay.
There's actually no significant difference there.
Yeah, but now it starts to differ, right? Because you have so many samples. So one thing you have mentioned is clustering, which is great, right?
Well, I mean that's what happened afterwards, right? Mm-hmm. Then analysis of this count table.
Yeah, exactly.
There are several things that we do there that are very different. Well, you can also do it with the bulk RNA sequencing, but with the single cell RNA sequencing, these things are way more. They are crucial. Everyone does that. Because that's what was the question of the experiment.
So apart from clustering, I imagine you may do some kind of principal component analysis. What else can you do with this data?
Well, the PCA and clustering are kind of two power horses. And obviously, they can be done based on like all the genes. But what is more useful is first you determine which genes are highly variable. And then you only focus on these genes for the further analysis, because this way you kind of increase your signal because you don't take all those housekeeping genes, all those things that don't really change. So that's kind of important. And to identify the highly variable genes, there are different methods. The simplest one is to look at the coefficient of variation of the gene's expression, and then you just pick those who are having high coefficient of variation, and then you analyze these. Because if the variation is high, then there's a big chance that actually there was difference. They are differentially expressed between clusters, for example. So that's something important. And then it depends on your biological problem. But for example, if you look at some dynamic difference, what you do is you want to order your cells according to this process. And again, there are thousands, maybe not thousands, but there are probably 10 or more papers already with different computational methods that deal with that. And the simplest one, the first one, was this one called Monocle, which basically uses first dimensionality reduction with ICA, and then uses minimal spanning tree to draw a trajectory from one state to another. But now there are other methods using way more sophisticated algorithms. Like my colleague from my lab, from my former lab, he used this Gaussian processes, latent variable models for doing the same. So it's good to just look through papers and see what's out there and what's like your choice. So this is an important problem to solve if you have this data.
Okay, so I'm curious how you treat this software. And of course, there is constantly new software. Before this software comes out, do you have a problem that you wish there was software that solved it? Or is it more like the software comes out and you're like, oh, wow, now I can do also this kind of analysis that I didn't think about before?
Well, when I started with the single-cell RNA-seq, there was no single-cell RNA specific tools at all. So then we had to figure out things ourselves, mostly because single-cell RNA sequencing community is quite tied together. So people would like talk with each other and on the conferences, and these things were kind of advancing. But I think the way the things were working was kind of together. Questions or like ideas, what we can additionally get from this data were being asked and the tools were being developed in the same time. I was in the lab and I was in an environment where, like previously, where most of the people or many people could develop these tools and they were interested in development of these tools. But it's not always like that, right? There are many labs where people are interested in, rather than on development of tools, they're interested in answering particular biological question. And where are we now with the single-cell RNA sequencing? Anyone can basically now use it. And these people would rather use tools than figure out things themselves. And they would just check different tools and see what they offer.
But do you think the community needs more tools? Do you have anything specific in mind?
I mean, I think most of the key problems are being addressed. But obviously, everything needs to be improved. And one of the big problems in this data is the problem of zeros. In normal RNA sequencing, if you sequence deeply enough, you will have no zeros basically in the count table. But the amount of zeros there is quite low, and if you have zero, it really means zero. While here, because of the sampling problem in the preparation of the libraries. So when you do, especially when you do reverse transcription, you may have RNA there, but you may still get zero because you lose quite a lot of material, and because there's so little to start from, basically. But in proportion, you lose quite a lot, 90%, around 90%. So, and that zeros are, you actually don't know whether in some place there is zero or actually there is some expression. So this is something that's important for the analysis because it's missing values basically. So this is a problem to be addressed, and there are already attempts and there are tools that try to input to infer this data. So there are ways there are methods being developed. Could be nice if there would be more.
Yeah. So it's not so much about the software, about the missing software, rather than missing methods or some statistical algorithms or models.
I mean, depends what people, like which scientists you want to address. Because if you think of people who actually do bioinformatics, they can use all the tools that are available and it's good. But there are not that many tools that are available to people who don't use the command line. And there is some room for development there, and they are showing up slowly. But that's something that we need.
Wait, are you saying that there are people who don't use the command line?
Of course, there are many biologists that are more interested in finding a new collagenase that will dissociate their cells, which is actually very difficult. So, they prefer to use things like tools that are kind of a bit more easy, kind of with the graphical user interface. Yeah, that's the real world.
All right. Now we've got quite a few questions from our listeners. This time I did an experiment. It was suggested by one of our listeners. And the experiment was to ask our listeners something that they would like to ask you, Aleksandra. And I will start with the questions posted on our website, bioinformatics.chat. So Alina asks, would be nice to hear about pros and cons of DropSeq and if there are other similar promising protocols. And I think you mentioned Dropseq somewhat, right? But maybe explain a bit more what it is and what the pros and cons are.
Well, Dropseq is one of these methods that is based on encapsulation of cells into droplets in the O phase. And there are many more of protocols like that being developed. So the other one that was published actually at the same time is InDrop by Alon Klein and David Weitz. So these were developed they were published in the same issue of Cell. But they are now also commercial solutions where you basically buy a box and everything is ready. Because for Dropseq, the problem could be that you need to assemble it yourself and you need to have some engineering skills to do that. But now, for example, there is this Chromium 10X from 10X Genomics. It's very similar in a way that you can buy. And there are many other companies that are either released or are going to release soon similar machines. The most important pro of these machines is that you can do a lot of cells at the same time. And that's great. The cons would be that it can be pro or con that it's a molecule counting method. So you don't get the full length. So you cannot do alternative splicing analysis, or you cannot look at the single nucleotide polymorphism throughout the RNA. So these are some cons. And the con may be the fact that it's really expensive as well. But you get a lot of data. So, the price per cell is low, but the price per bulk is high. So if you don't need to have thousands of cells, if you only have a couple hundreds, then it could be expensive.
And so the price here is driven by reagents?
By sequencing.
Okay.
Because if you have thousands of cells, you need to sequence them.
Fair enough, yeah.
And the way it works is it's that you cannot, out of this pool of cells where you have several thousand, so let's say between 2 and 10,000 or about 2 and 8,000 cells in these technologies, it's not that you can take 300 cells apart and only sequence 300. No, you have to sequence the whole thing because everything is pulled together. You could lower down the number of cells by giving the lower density of cells into the machine. But then again, the price of cells will rise because you use as much as reagents. So, you have to fine tune which method to use, like price wise, depending how many cells you want. There was recently a very good review. Unfortunately, I don't remember an author, I think in Genome Biology, where the authors compared all the different methods and they also gave exact prices. Maybe I can look for that and put a link somewhere.
Yeah, awesome. Ming-Fu Shao, who, by the way, was our guest in our previous podcast, asks, I particularly would like to know the difference between regular RNA-seq data and single-cell RNA-seq data. And I think we discussed one difference already, which is sometimes single-cell RNA-seq data, it is sequenced just from one end, right, to do some crude counting.
Mm-hmm. Well, you can also do that with normal regular RNA sequencing, actually.
Yeah, right. Although I don't think it is done much nowadays, but maybe I just don't know.
It's not, but it can be.
Right, yeah. So do you know of any other differences between these two types of data?
Well, the number of detected genes, right, but is very different. So in the regular RNA-seq, you detect way more genes, and the precision of your measurement, like sensitivity, is way higher in comparison to single-cell RNA-seq. But with the single-cell RNA-seq, obviously, what you gain is the information on the single-cell level, which you cannot ever get with regular RNA sequencing. So this is like a conceptual big difference. But other than that, one difference that maybe is not so often shown is because you do a lot of PCR or a lot of amplification steps in the single-cell RNA-seq in comparison to regular RNA-seq, you introduce some errors while you do PCR, like base errors. So when you want to use this method for looking at single nucleotide polymorphisms, it is not ideal because of that.
Yeah. On the other hand, if you want to resolve single nucleotide polymorphisms, you can use all the cells together, right? Because they share, well, like 99.99% of their DNA.
Well, of course you can do that, but let's say if you had this study where you look at sperm cells and you want to assess the difference between sperm cells because they have different how evolutionarily, it's not, maybe not evolutionary, but how they differ from each other, and then for this kind of analysis, if you wanted to do it on RNA level, which is still probably a bit easier than on DNA level at the moment, you couldn't, it would be difficult to find, for example, de novo mutations.
Oh yeah, I see.
Right? Not things that are there from the beginning, but, because these maybe are not so interesting, but maybe some de novo things where you could kind of build a tree of mutations. People are thinking about this, but at the moment, technology is not yet there to be able to do that, but it would be super interesting.
Mm-hmm. Okay. And now we have quite a few questions on Reddit. So user Bucaro asks, library preparation techniques, advantages and disadvantages of using molecular counting or SMRT or something else.
Okay, so this person just asked about some particular protocols. The molecular counting, as we already discussed, it's really useful because you avoid the PCR bias. So you really know how many molecules you have. But again, as we discussed, you only have the end fragment and only know how many molecules. You have no idea about what kind of alternative splice variants you have in your sample. So you cannot look at that. So if this is interesting for you. And another advantage of the end counting is it's cheaper to sequence because you only sequence the end of the molecule. All these methods have advantages and disadvantages in general. And I would really refer to there are two recent papers that reviewed this problem, compared different methods in a systematic, good way. And let me check. I'll find it.
We'll just post all the links that you provide to the website. They'll be in our show notes. Awesome. User Enhancer RNA, that's their username, asks, basic workflows. Never worked with single-cell RNA-seq, so it would be nice to learn about what packages for what and the advantages versus disadvantages of those packages at the moment. So I think the user asks about maybe R packages or something like that.
Mm-hmm, mm-hmm. Well, at the moment, there are many, and it really depends what this person wants to do. I'm not a bioinformatician by training, so I don't really know things about comparing speed, for example, of the method and things like that.
Sure, but maybe you could talk about some of the packages that you used yourself in your analysis.
For example, as we already discussed, for looking at the trajectories, one of the simple packages that's very easy to use is a package called Monocle, which uses the ICA and then minimal spanning trees. It's really useful to look at the trajectories. Then the other one that we used is called SCLVM. And this package is, this does is basically when you have a heterogeneous population, the heterogeneity comes from different things. It can, for example, come from cell cycle and from the differentiation of the cells and from something else. And basically what this software does, it enables to disentangle these different processes. So, for example, the signal of heterogeneity from cell cycle usually is one of the most overpowering in your analysis. And if you put your cells on the PCA, you will not see anything. But once you remove the signal from the cell cycle using this method, what you can do is you can then put this data to, for example, do clustering or participant component analysis or something else, and it may actually show some good results that was not able to show before. So that's another package. There's another package, for example, called Cyclone, which enables to where you give it the data and it tells which cell cycle stage the cells come from, which is also nice. There are so many, really.
So I'm wondering, how is this done? Like when you're trying to resolve the phase of the cell cycle, is it that there are some well-known genes that are responsible for each phase?
Yes. So there is a database called cyclebase.org, which is really good. And they have experimental data from all different genes. And they can see which of these genes are expressed specifically in a particular cell cycle stage. And based on these data, as well as on the data that were generated, like from cells that were cell cycle stage was known, because they were stained, so you can understand from which cell cycle stage they are. The guy who developed it, Antonio, he was able to learn a model that kind of can tell which stage of cell cycle the cells come from. The other things that people use is it's really so many. So there's, for example, from the lab of Dana Per, there are several different methods that work on the pseudotime ordering, which are very good, and also on clustering. So that these are also useful to check out. Although some of these were developed for mass cytometry, they also can work for single cell RNA-seq.
Cool. User not citing lamely asks, well, there are two questions. First, about costs regarding barcode library diversity. And I think you mentioned a paper that gives some cost estimates. So I think we'll locate.
Yeah. I will give this reference to you. Mm-hmm.
Yeah. Okay. And the second question is transcriptional artifacts caused by cell isolation selection methods.
Well, that's a very good question. It's true. I mean, cells get stressed when they are isolated from the tissue, especially if the tissue is being treated, for example, with some enzymes like proteases to break the structure. And they are not the same as if they were in the tissue. It's obvious. This can be minimized by doing it quickly and in a cold temperature, but it's going to be there. So there are some methods being developed of looking at the single cells, or at their transcriptomes in tissues using microscopic-based methods. And I think these two are complementary. So if you want to make you can discover many things using single cell RNA-seq, because some of the signals are still going to be there, obviously. But if you are worried about effects coming from the stress of extraction of the cells, then these methods could be a solution.
And in terms of protocols, are there protocols that are more sort of gentle in this regard than others?
Well, I mean, it's not about single cell protocols per se that are gentle or not gentle. The problem is the isolation of cells, which has to be done exactly the same way for many other types of experiments. So it's the moment when let's say you want to have some particular cell type from kidney, you need to get a piece of kidney. And if it's a piece of kidney from a patient, then first you need to go to the hospital, pick a kidney and travel with it to the lab. And then you need to break the kidney either mechanically or enzymatically, and this takes time and the cells are stressed. And this is not related to single cell sequencing, actually. It's just you need to get a single cell, and that's the problem.
Right, but that's a part of the protocol, isn't it? So, I imagine there are different methods to isolate cells, and some of them may be better in this regard.
Well, it depends again on the cell type, right? Because if you want to extract some cells from the skin, it's different than if you want to get blood. Because blood you just get, you don't have to put any enzymes. The cells are already natural single cell. Or the spleen, it's enough to smash it a little bit with something, just press it and you get the cells. But if you imagine skin, it's a very tough tissue. So it takes a lot of enzymes to digest it and so on. So this is specific to the tissue of interest rather than the protocol. So depending on what tissue you use, there are different methods.
Okay. User Moody Stocking asks, my research is on single-cell RNA-seq, so I would recommend including something about the technical noise that comes with the single cells and the best ways to deal with this.
Yes, so it's true. There's technical noise that is way more pronounced than in the normal standard RNA-seq.
What are the main sources of this technical noise?
They come from the fact that you have a low amount of material. For example, when you sample with the reverse transcription, your enzyme is going to hit only some of the molecules, so you have some noise already at this stage. The noise basically comes from the biochemistry of the preparation of the library. And it's more pronounced because you have very little amounts of the starting material. And the best ways to deal with it, I think, is always to add a spike-in, because spike-in will be subjected to all of the processes that your sample goes through. And the spike-in, you know how much you have there. So all the noise and all the heterogeneity that you will see in a spike-in, that is, all the heterogeneity of the spike-in is kind of your baseline heterogeneity. So everything that is more heterogeneous than your heterogeneity of your spike-in will be actually genuine heterogeneity, genuine differences between populations. So that's why spike-in is important. And then the other thing is, I would totally recommend to repeat the experiment. So if you have, let's say, 3 different conditions, so you do it on one day and then you do it again on the same day, if you see the same result, and that results that will observe that what you observe is not coming from the noise. And
speaking of spike-ins, you say that they're great to combat this technical noise.
Well, to measure it,
yeah,
we cannot really combat it, but we can know how much of the noise we have, which is already useful.
Right. On the other hand, well, there is still some stage at which you introduce the spike-ins, right? So, for instance, what you described about the stress on the cells, right? Presumably, you cannot introduce the spike-ins as early as that.
No, you introduce at the stage of lysis.
Yeah. So whatever happens before that, you cannot measure that.
But this noise that you are describing is a biological noise. It's a biological difference because cells get stressed, but what happens there, it's a biological process. Mm-hmm. So spike-ins can control for technical noise, the noise that comes from the process of the library preparation. And they also are not perfect because they don't control for the lysis of the cells because we add it to the lysis buffer. So because when you lyse the cell, what can happen is that not all of the molecules, the lysis doesn't happen properly, and not all of the molecules are released to the solution. So it doesn't control for that. But at the moment, this is the best we have.
Right. User Sanger Sequence asks, any thoughts on how big of a problem Illumina's index swap problem with the XAMP really is? And they link to an article which I believe is...
I know.
Oh,
I know this article. I mean, I haven't read it. I read the abstracts, but I can... it is an issue, obviously, if it's as pronounced as the authors describe in the paper. So one of the solutions that we could do is to have a barcode on both sides of the molecules, a unique barcode, right? So this way, when you flip, you'll still know by reading the other barcode which molecule it is. So that could be a solution. Mm-hmm. But the other solution is to go back to NextSeq, so the old chemistry, or use HiSeq 2000. But, well, I'm sure this problem will be resolved quickly.
Okay, let's hope. User Organelas asks, capture efficiency. How much information are we missing?
A lot. I mean, there are some estimates that 90% will miss. It slightly depends also on the method. Some methods capture a bit more than the others, but yeah, it's probably around 90% that we are missing. And I think this may be slightly improved with better enzymes, but we will always miss quite a lot, probably.
Mm-hmm. User Whom asks different ways of accounting for a cell cycle and how to correct for it.
Oh, yeah, that's my favorite cell cycle. So the first way is to use the method that I described, the SCLVM method, and basically regress the effect of cell cycle from your sample. It works very well, and I really recommend it. The other way, what you can do is you can assign cell cycle stages using methods like OSCOPE, or you can cluster cells using cell cycle stage specific genes from the cyclebase.org database. And then using that, once you know from which cell cycle phase your cells are, you can only compare the cells from the same cell cycle stage. That's another option you could try. So at the moment, I think these are two ways people dealt with the cell cycle issue. But the SCLVM, it probably is the superior way if you just want to remove the cell cycle effects from the data.
Mm-hmm. User Cyrus Base VI or something like that asks actually 4 questions. The first one is how to detect and deal with batch effects.
With spike-ins. So when you sequence your samples, and if you have spike-ins, what you can do is you can extract the spike-in and try to cluster on the spike-in only. And you will see if the spike-in clusters in the batches, then there's something wrong and there's some problem. Mm. And the other way is to do a biological replicate of the experiment. So basically do the same experiment again and again, and then you will know.
Yeah, especially like if you plan to publish in Nature, maybe try to replicate your experiment yourself, right, before someone else does it.
Yeah, no, definitely it's useful to do the replications.
Yeah. A second question is best way to deal with dropouts. And I'm not sure if we have defined this term so far. So what are dropouts?
Mm-hmm. So dropouts are these zero measurements that I mentioned. So it's in the case where the gene is there, but we didn't detect it. So we observe zero, but we measure zero, but that zero doesn't mean that the molecule wasn't there. It's a tricky problem, and that's what I was discussing before, that this is something that is being worked on. I mean, the easiest way is to disregard of lowly expressed genes, and then this problem is avoided. Because if you have genes that are highly expressed, this problem is not visible. There are some methods that looked at that. One of them was called I think it's called ZIFA. They looked at that, but I'm sure that there will be more appearing.
Yeah, this goes back to this idea of borrowing strength. So if you see that some gene or some isoform is well expressed, or more or less uniformly perhaps expressed in like most of the cells, but absent in some of them, then using some kind of probabilistic model, you can conclude that, maybe it's absent from this particular cell, but there is also good probability that it is present. It was just not sequenced.
Yes, but I think it's a little bit more difficult in this case, because what you will see will be, it won't be that majority has it and just one doesn't. It would be minority has it and majority doesn't, and from this you would try to impute. Because if you lose 90%, then most of them actually won't detect. If you have something with a low expression. So, I mean, it depends on what expression level you want to look at. What you described is probably the situation that you could use with genes that are moderately expressed. But for genes that are lowly expressed, it's more tricky.
Mm-hmm. Next question is there a way to do power analysis for single cells?
Yeah, and I can. There is a paper by my former colleague Valentin Svensson in Nature Methods.
I saw that, yeah.
So yes, there's a way, and it was done.
Yeah, but it's interesting in which sense they use power analysis, because the word power, I think, literally occurs, like apart from the title, it occurs maybe 1 or 2 times in the paper itself. So, like the way, at least I'm used to think about power analysis is when you have maybe some statistical hypothesis, right? You have like a specific analysis that you have in mind. And then in order to maybe reject the null hypothesis, you do this power analysis. But I think in the paper you mentioned, they talk more about like general sensitivity and precision of measuring, the expression.
Well, but he also looks at, for example, how deeply you need to sequence to get information. So basically, like if you sequence more, then you don't get any more like useful information.
Interesting. So there's like a point of saturation there.
Yeah, that's a big difference between this the normal RNA-seq and this. That here, if you sequence more after some moment, it doesn't matter because you just resequence the same thing. So this thing is also in that Nature Methods paper that I'm sure we will put a link to.
Yeah, absolutely. And I think the last question is good packages for differential expression analysis. So I imagine they're more or less the same as for the classical RNA-seq, or are there any single-cell specific packages?
There are some single-cell specific packages as well. It depends how you look at it. You can, if you want to compare between clusters, and you want to assume that each cell within a cluster is a replicate, then you can use the standard methods that are used for standard bulk RNA sequencing, like DSeq. But there are some methods that are specific for single RNA-seq, but I don't remember the name, unfortunately, now. Like, this is not something that is so much used, but because the way we analyze this data is rather looking at which genes are heterogeneous, and then these are kind of differentially expressed between the clusters. But we first identify heterogeneity rather than doing differential expression, if you understand what I mean. But there are some packages. At the moment, it's just not on top of my head.
All right, great. Now, before we finish, is there anything you would like to talk about or to tell our listeners, to promote anything?
Well, I think if someone thinks that that is a complicated thing to do in the lab, I would like to encourage people and say that doing single cell RNA sequencing is pretty easy, and you can really do it in any lab. It's nothing to be afraid of. And now, because the community is really large, there's lots of people you can ask how to solve different problems. And definitely, I would like to recommend, if someone is interested, to come to our yearly single-cell genomic conferences. And this year it's in Weizmann Institute, but it travels around Europe. So I totally recommend that, because it's a good source of knowledge about different packages and different methods, and both computational and wet lab. So if someone wants to get more information, that's definitely a place to go.
Awesome. Well, Aleksandra, thanks a lot. This was a very educational conversation for me, and I hope for our listeners as well.
Thank you.